Hello everyone, my name is Wesley and this video is my lab final in Biological Science 214 Microbiology. During this final we were required to do a lab simulation called Save the Patient, in which an unknown bacteria was assigned to our patient, and through a series of tests we were to isolate this bacteria from a list of unknowns given to us prior to the start of this event. Now, not only was this required just for a grade, but it was to demonstrate what would happen in a hospital lab setting. So a patient would come in with symptoms and it would be caused by a bacteria that we wouldn't quite know. And through these series of tests that I performed, we would be able to isolate that bacteria and effectively and properly prescribe them the correct antibiotic. So this lab simulation was very important to teach us the steps that are going to be taken. So I really enjoyed going through this because I was able to demonstrate my knowledge of the test, and it confirmed that I knew kind of what I was doing. So the tests that I was able to do during this were a gram stain, an Endall test, a citrate utilization test, and a hydrogen sulfide production test through a test called TSI. So let's talk about gram stain. We've talked about it before in the past, but let's go over it once again. The purpose of the gram stain is to determine the reaction towards the bacteria from the gram staining. It'll tell us whether the bacteria is a gram positive or a gram negative. Now, the gram stain uses 4 reagents. It uses crystal violet, iodine, a decolorizer, which is normally alcohol, and safranin, which is a red stain. So the crystal violet will dye all the bacteria a purple color, and then the iodine will come in and it will begin to break down the plasma membrane, and the decolorizer will come in, the alcohol, and it will strip the gram-negative bacteria of its purple color, making it clear almost. And then finally you'll have the safranin stain that will come in, and it will dye all the gram-negative bacteria a pinkish red color. And that's how we do a gram stain. And that, again, will tell us whether it's gram-positive or gram-negative, and in turn will tell us which set of tests we should do after that to determine which bacteria it is. Next, I did an Endall test. An Endall test is used to determine the ability of the organisms to split indole from tryptophan, and it uses the Kovac reagent. Now, the Kovac will react to whatever the bacteria is doing inside this test, and you're either going to get a red ring at the top of your broth, which would indicate that indole is effectively split from tryptophan, or you'll get a yellow ring, which will indicate that indole is not split from tryptophan. And again, this will determine which test you'll do after that. My next test that I did was a citrate utilization test, and this is to determine whether or not the microbe will be able to use citrate as a source of carbon, which would be used for energy and to allow it to grow. There are no reagents that are used in this test, and the results of the test will determine whether or not the original green color of the agar solution changes to blue. So if it changes to blue, it's a positive test, which means citrate is being used by the bacteria as a source of energy, as a carbon source. And if it stays green, it means that citrate is not being used. Now, the last test that I did to determine my unknown bacteria was a hydrogen sulfide production test, which is done through a TSIA. Now, what that is, the TSIA actually gives us 4 results. It will tell us whether or not the bacteria uses a glucose or lactose or sucrose as energy. It will tell us whether or not acid is being produced. It'll also tell us if gas is being produced. And it will, again, which is what I used it for, tell us if hydrogen sulfide is being produced by the bacteria. The reason why I chose to do the TSIA was because, 1, it's used for gram-negative bacteria, and 2, I needed to know whether or not my bacteria produce hydrogen sulfide or not. So, as you can guess, the purpose of the hydrogen sulfide portion of TSIA is to determine if the microbe reduces sulfur-containing compounds to sulfides during the process of metabolism. There are no reagents that are used in this, and what I looked for was to see if the original red color of the agar changed to black. And if it did, that means hydrogen sulfide is being produced. If it did not, then it means hydrogen sulfide is not being produced. So those were the 4 tests that I did. Those were the purpose of those tests, the reagents that were used, and the observations that we're looking for, whether it's going to be positive or negative. So let's talk about how I went through the process of eliminating my bacteria. So I'm going to read the list of bacteria to you. There are 12 of them in total. We have the Enterococcus avium, the Enterococcus faecalis, the Escherichia coli, the Klebsiella pneumoniae, subspecies Ozaniae. We have the Cocrea rosea, Proteus vulgaris, Providencia stuarti, Salmonella enterica, Staphylococcus aureus, Staphylococcus epidermidis, Streptococcus sanguinis, and Yersinia pseudotuberculosis. That is a mouthful. So at the very beginning of this lab simulation, we are given a gram stain reaction. So this is how we determine whether or not it's going to be gram positive or gram negative. So my gram stain came back as a gram negative rye. So my gram stain was either gram positive or gram negative, and my result was gram negative. So based off this one test, we were able to split our unknowns into a positive category and a negative category. So the positive category, I didn't worry about anymore because my bacteria is a gram negative. So I went from a list of 12 unknown bacteria down to just 5, possibly 6. We have the Escherichia coli or E. coli. We have Klebsiella pneumoniae, subspecies Ozaniae, Proteus vulgaris, Providencia stuarti, Salmonella enterica, and the Yersinia pseudotuberculosis. Now, because my gram stain was negative, I had a specific item or a specific list of tests to perform. So it's called the MVIC series, which is your indol, methyl red, Vos proscure, and citrate. So in this lab simulation, and even in the real world, you're given what could be called an identification matrix. That's going to tell you whether or not the bacteria you have is going to have a positive result, if they generally have positives, if they generally have negatives, or if they're somewhere in the middle. Using that identification matrix, I was able to look at my bacteria that I had and determine which test would be best to go with next. And that's how I chose the indol, because it looked like indol would split them about equally. So after my indol test, I noticed that there was a yellow ring at the top of the broth. So that told me that in an indol test, my bacteria is negative. So that again split my unknown bacteria into a positive category and a negative category. Because my results were negative, I did not worry about the positive side going forward. But I was left with my Klebsiella pneumoniae subspecies Ozaniae, my Salmonella enterica, and the Yersinia pseudotuberculosis. So looking at the list of tests that I needed to perform for a gram-negative bacteria, and then what would also differentiate my bacteria even further, I chose to do a citrate test. So I wanted to know whether or not my bacteria used citrate as a carbon source of energy, or if it didn't. Because 2 of them did, and 1 of them did not. So my citrate test changed from green to blue, which means that it did use citrate as a carbon source. It's a positive test. So I was able to take those 3 bacteria, and now I'm left with Klebsiella pneumoniae subspecies Ozaniae and a Salmonella enterica. So now look at those 2. Those 2 bacteria have a very distinct difference. Salmonella enterica produces hydrogen sulfide as a byproduct, so it can reduce sulfur containing compounds to sulfides. And then Klebsiella pneumoniae subspecies Ozaniae, it does not. So I knew that by doing a TSI-A test that I would be able to differentiate between the 2 unknown bacteria that I had. Now, at this point, I'm on day 2 because I did my gram stain, I did my indol test, and I did my citrate test all on the same day in this lab setting. So I only have 1 more day to perform a test. So thankfully, I narrowed it down to just 2 unknown bacteria and was able to differentiate them using just 1 test. So I did my TSI-A, and I was looking specifically to see if it produced a hydrogen sulfide or not. So I did the TSI test, which is done by, much like the other two tests, sterilizing your inoculating tool, which in this case is a wire, and transferring bacteria from your culture into your TSI-A test tube, which is your agar solution containing all your compounds for TSI-A. And then it's incubated overnight at 37 degrees Celsius, and the next day you're gonna look for all these different changes. So that's what I did. I transferred my bacteria aseptically from my culture into my TSI tube, incubated it overnight virtually in the 37-degree incubator, and upon looking at the test, I was just looking to see if it changed from red to black. And it did not change from red to black. It was yellow, it was broken up, but it did not change to black, which told me that it did not produce hydrogen sulfide. So I went back to my identification matrix, and there was only one unknown bacteria that was left, which was the Klebsiella pneumoniae subspecies ozanine. And thankfully, I did everything correct, and that was my unknown bacteria. So this is my lab final video for Biological Science 214 microbiology class. Thank you so much for watching my lab videos in the past. I hope you enjoyed watching what I had to do for my final, and I really enjoyed this class. So thank you again for watching, and enjoy the rest of your day.